my name is Patricia Cabrera and I work for the Flanders Marine Institute in Belgium. I am demonstrator leader for the zoo and phytoplankton EOVs, or essential ocean variable products. And in this presentation, I'm going to show you how to navigate this demo and what is the connection with the Blue-Cloud Hackathon. So for the topic, understanding the ocean, and the challenge 1A, mapping hotspot of plankton diversity, it will be very useful to get acquainted with this demonstrator. You could answer questions like how might we map hotspot for plankton diversity and identify geographic gaps in our knowledge using currently available plankton datasets. So for this, I will show you what are the products that we have developed, which consist in three different parts. We have Phytoplankton EOV products, Zooplankton EOV products, and modeling phyto and zooplankton interactions. I will finalize the presentation by giving a live demo on navigating in the virtual research environment in Blue-Cloud. So first, the phytoplankton essential ocean variable product consists on two different products. The first one is a product that has been developed to generate global ocean 3D chlorophyll A concentration maps. This is a proxy of phytoplankton biomass, and this has been done by using machine learning techniques. So for this, we can see a set of input data in this box where we have used Argo and Copernicus products like biogeochemical Argo floats for the chlorophyll A concentration, satellite-derived reflectances, satellite-derived photosynthetically available radiation or PAR, sea level anomaly, and temperature salinity profiles. So for this, all these datasets have been applied to a neural network model, what we are calling here the chlorophyll A model, that has been developed based on the methods based on Sauzade et al. 2016. And this is presented in a Jupyter notebook. Now here on the graph on the right, we can observe the main output of the model. So we can see a latitudinal gradient and a decrease of chlorophyll A concentration with depths, which is, if you see in the GIF, we have 36 different depths. So this product have been developed by our colleagues in the Laboratoire Oceanographique Villafranche. And the second product consists with the same input data, but here a second neural network model was trained, what we are calling the PFT model. So these are generating global ocean phytoplankton functional type products. And again, here on the right, we have a map with the main output of the model. So here we see again representing January for the months of winter, and 36 depths. So this figure, we can observe a latitudinal gradient from a small to larger phytoplankton groups from the bottom of the graph to the upper part. And the latitudinal gradient we can observe, it goes from the equator towards the poles. Now, the second product that was developed in this demonstrator, it consists of zooplankton distribution gridded maps. So for this, we have several input data. We have zooplankton abundances from the CPR data from the Marine Biological Association that can be accessed through EMONET Biology, and now as well from Blue-Cloud. There is also several environmental data like temperature salinity climatologies from CDataNet, also accessible from Blue-Cloud, nutrients from the World Ocean Atlas, and the distance from the coast from NOAA, and bathymetry from JEPCO. So this was applied variational analysis and neural networks. And using the DIVA-ND tool and a neural network model, we have generated maps of zooplankton distribution in the Northeast Atlantic region. So what we observe here in the panels is one of the distribution of one of the most copypot species, most abundant copypot species. And we can see how we can go from heterogeneous observations on the panels on the left to a more homogeneous gridded fields in the panels on the right. This is also presented in the virtual research environment in Jupyter notebooks, and this is written in Julia and has been developed by our colleagues in the Liège University. Now, the last product is a model which we're calling the model phyto and zooplankton interactions. And it helps us to understand what are the drivers that limit phytoplankton abundance. So here we're using as well a several set of input data, which consist on zooplankton and phytoplankton abundances and environmental data, nutrients, temperature, and light. All this data can be accessed from LifeWatch Belgium and Emonet Biology, as well in Blue-Cloud. So the model developed here is called the nutrient phytoplankton zooplankton model, which is based on the model already developed by Soetart in 2009. And on the right, we can observe one of the main outputs of this model, where we see what are the relative contributions that drive phytoplankton abundance. This data is being used for the Belgian part of the North Sea. And what we observe here is that zooplankton grazing and light are the main drivers, followed by nutrients and temperature. These products have been developed by our colleagues in the Flanders Marine Institute. So now, how to access the VLab? So here in this link, you can directly access our VLab, the zoo and phytoplankton EOV products VLab, as you can see here in the computer picture. And then after you have registered, you will land in our VLab landing page that I'm going to show you in a video after this slide. But first, I would like you to keep in mind that all the information to know well about the demonstrator is captured in the handbook. This will be available for you to check. But also there has been a starter kit for you with extra resources to be able to use this. And as well, some important aspects is that you will always have to work in your own workspace, but all the documents are placed in a shared workspace that it is now read only, so there is no risk of deleting or changing the scripts. Now, if I just show you how to access the demo, voila. So once you have registered, you will see a page like this, and then you will log in. So apart from all the resources I have showed you before, we also have how-tos. So here is an example of the how-to on how to run the ARC Markdown model for the NPZ model. And also you can run the Jupyter notebooks by first launching the Jupyter Hub. And here you go to the workspace. This is the shared workspace that I have mentioned before, where you will find all the documents needed. And it's very important that you first save these Jupyter notebooks in your own workspace. So here I'm showing you how to access the notebooks. And I have already copied them into my own workspace. And now I will just open the file and show you how to run the notebook. So you first need to run all cells. Once you have done this, you will notice that here on the upper right corner, we see how there is a gray dot. This means the notebook is running. As long as this is running, you will see always this in gray. And when it's been already run, we can see the output. So for the purpose of efficiency in this video, I have already run the notebooks, and I'm showing you how after each cell, you see in pink how is the output. So for the zooplankton products, you can see that there is 2 notebooks. This is the first notebook that has been run, and the results have been saved. And then we need to plot the results. So for this, we have a second Jupyter notebook that I'm showing you now, and that again, it has been already run to show you already here the results, the gridded maps I have shown in the first slide. So this is all for running the Jupyter notebook and the R markdowns, which is the main component of our VLab. And again, for any doubt that you may have, you can always contact us and also read further the handbook and the starter kit. So as well, you can find some mentors that we all have different expertise. Mainly for any questions for this demo, you can contact me as the first contact, but as well, there will be Charles Trupan, which is who developed the DivineD product, and as well Leonard Heppers, who is very aware of the functioning of this demonstrator. We also have some other mentors that can help you with how to find data in LiveWatch, Eurobis, and this is actually what we want. We want that you develop further these products to answer the questions of this challenge and this topic. And for this, we are here to help you in anything you may need. So good luck, and we are very excited to see what you can do with our products. Thank you.